Morning everyone. This class is about cell division, that is meiosis. In the last class we have seen the mitosis, and in this class we are going to see meiosis. See, as the NEET exam nearing, we will see the chapters very quickly. So follow this class continuously and get good marks. Okay. All the best to all the students, those who are appearing for the NEET examination 2025. Okay, let us go into the class. See, in this class what we are going to see is the 5 phases based on chromosomal behavior. That is in Preface 1 you can see 5 different phases. Okay, and meiosis 1, meiosis 2, crossing over, how the crossing over occurs and the importance of crossing over, that is very important. So that we are going to see in this class, as well as the significance of meiosis. Okay, meiosis 1. Meiosis 1, in the sense, meiosis takes place in 2 different cell division. Okay, meiosis 1 and meiosis 2. So in meiosis 1, the prophase 1, this is very important and very interesting thing to learn. Okay, how this occur. So prophase 1 occur in 5 different phases. One is leptotin, next one is zygotin, packaging, diplotin and diakinesis. So what is happening in this, we are going to see. See, I told you the first phase is leptotin, second one is zygotin, third one is packaging, fourth one is diplotin and the diakinesis. Okay, what is happening at these stages in the sense? See, in leptotin you can see this is the cell and the cytoplasm, and you can see the nucleus. And I already told you in the mitosis there are 23 pairs of chromosomes are present in which cell? The somatic cell, isn't it? How many? Let me take the pointer. How many chromosomes are there in the sense? There are 23 pairs, okay, in the nucleus. 23 pair in the sense, 23 pairs in the sense, 23 from the father and 23 are from the mother. That is the single pair. See, for example, this is the pair in the sense, this pair is called as homologous chromosome. Why it is called as homologous chromosome in the sense? The shape as well as size are same, okay, but one is from father and another one is from mother. Understand? So this is called as homologous chromosome. Why it is called as homologous chromosome in the sense? Both are in the same size and same structure. Okay, so it is called as homologous. So what is happening here? I told you already 23, but for the easy to observe and easy to study, we have taken only 2 chromosome here. Okay, so see what is happening at the stage of leptotin in the sense, these sister chromatids are coming together. Okay, and sister that is the chromosome is divided into chromatids. Okay, these are called as sister chromatids. And in the zygotin stage, what is happening in the sense, the chromosome are coming together. Okay, coming together closer. So homologous chromosomes, these are called as, they are coming together. And here what it is called, this is called as non-sister chromatid in the sense, the crossing over takes place at this packaging stage. Okay, so packaging, what is packaging in the sense? See, this is homologous chromosome, isn't it? The homologous chromosome, they have the chromatids. So these sister chromatids, here what you are saying is non-sister chromatid, in the sense one chromatid from this chromosome and another chromatid from this chromosome, there is a crossing over occurs. Okay, crossing over in the sense, how we can see this crossing over? See, you can see clearly here, this is the homologous chromosome. You can find the same size, isn't it? Same size as well as the structure is same. but the genetic materials are different. That is the genetic material from father, and this is completely different from the mother chromosome. Okay, genetic material. So now what is happening here? The chromatid formation, this is called as sister chromatids, and these two are also sister chromatids of this chromosome. Okay, here what is happening? This chromatid is coming here, and this chromatid, there is a crossing over occur. Okay, so at this stage what is happening in the sense, there is a separation. That is how it separate in the sense, this chromosome separate and this part is united with this chromosome. Got it? Got it? And the red, this red, red, then again it's with the blue. Okay, so this way this sister chromatid got the red part and this got the blue part. So now you just imagine, father is of one genetic character and with the facial and as well as the body structure. Mother is with another facial pattern and the body structure, but the child is having, there is no similarity between the, very few similarities are in the, you can find in the child, very few similarities you can find in the child. That is color color combination. Sometimes both are very fair, the child is very dark. Okay, and sometimes both the parents are very beautiful, the child is not that much beautiful. Sometimes you can take the hair complexion of the mother and the father, in the sense it won't be with the child. Okay, so from where this child get, in the sense, from his forefathers. Okay, from the forefathers of the mother as well as the forefathers of the father. Okay, that is what this genetic material, because of this genetic material changes, bring the child in a new pattern. Got it? Got it? So this crossing over is very, very important. This happens only in the meiosis division and not in the mitotic division. And also it happens only in the meiosis 1, not in the meiosis 2. Okay, so crossing over is very, very important, and where it takes place, in the sense, the stage of packaging of prophase 1 of meiosis 1. Got it? Got it? So meiosis 1, if you take meiosis 1, okay, meiosis 1 has prophase 1, metaphase 1, anaphase 1 and telophase 1. Okay, in the prophase 1 there are 5 different stages. What are those? Leptotin, zygotin, packaging and diplotin and dikinesis. And one more important point what you have to keep in mind, in the sense, on which stage of prophase 1 the crossing over occur, in the sense you have to answer it is packaging stage. Okay, and what is the importance of this crossing over, in the sense, exchange of genes from one chromosome to another chromosome, that is called as crossing over. Okay, so this is very, very important. That's why in human beings we can see the variations from forefathers to the grandparents and grandparents to the parents and from parents to the children. Okay, you can see this very clearly here. See, I told you the homologous chromosome, right? So this is, just imagine this chromosome is from mother and this chromosome, the blue color chromosome, is from father. Can you see this? The structure as well as the shape are very, very same, isn't it? The size as well as the structure are same. So this is called as homologous chromosome, and the center is called as kinetochore. Okay, this is kinetochore, in the sense it is, this point is called as centromere, and inside the centromere we can find the kinetochore. Okay, here only the separation takes place while the cell division, the spindle fibers attracts towards the opposite pole. Here only the separation takes place. Okay, and this already I told you, it is homologous chromosome, and these chromatids are called as sister chromatids. Okay, sister chromatid. And what is kinetochore? Kinetochore is a molecule which is present in the centromere. Okay, So I hope you understand the crossing over and the importance of the crossing over, as well as what is homologous chromosome. Homologous chromosome in the sense it's a pair of chromosome, one is from mother and one is from father. At the time of cell division they pair, okay, they pair, they come closure, and what happens there in between these homologous chromosomes, the crossing over occur. So during crossing over, the genetic materials are exchanged between the chromatids, okay, chromatids of homologous chromosome, not among the sister chromatid, okay, among the non-sister chromatids. And let us see what is metaphase 1. Metaphase, I already explained you what is metaphase, in the sense just like the mitosis, just like mitosis, the centrioles, okay, see, you find this is the centriole of the cells, in the sense they move towards the opposite pole, okay, they move towards the opposite pole, and they produce spindle fibers. What they produce? They produce the spindle fiber. These spindle fibers are very, very important at the time of cell division. Only at the time of cell division they form, okay, and they join with the centromere of the particular chromosome, okay, and at this stage I already told you they all come and arrange in the equatorial plane, isn't it? The chromosomes all arranged at the equatorial region and form a plate-like structure, and this spindle fiber joined at the centromere of each and every chromosome. So at the time of pulling, okay, these spindle fibers are contracted, okay, contract, or it is pulled from another side, in the sense what happened, the homologous chromosomes here, we have seen that homologous chromosome, right? So one of the chromosome moves towards that side and another is coming towards this side, okay. So the spindle fiber attracts the chromosome towards the opposite pole. So this stage is called as metaphase, okay, this stage is called as metaphase. And in metaphase 1, in the sense I told you, it is 23 pairs of chromosome, right? So 23 chromosomes are arranged like this, and another 23 are homologous chromosomes are arranged like this. Understand? Okay. Now metaphase 1 is this one. And what is anaphase? What is anaphase in the sense? Just take me another blank one. Okay. So what is anaphase 1? Anaphase 1 in the sense, already which one is arranged? Which one is arranged? The homologous chromosomes are arranged. Let me take only 4, okay. Homologous chromosomes are arranged here, we have seen, right? And now a pulling takes place. So due to pulling, due to the contraction, what is happening here, we will see what is happening here, in the sense the homologous chromosome, okay, the homologous chromosome, this is homologous chromosome 1, 2, 3, let us take, or 2, let us take. This is the contraction of spindle fiber, contraction of spindle fiber, that is the spindle fiber joined with the chromosome, not the chromatid, okay. So this is, just imagine 23 are here and another 23, so total how many? 46 chromosomes, okay, 46 chromosome. So these spindle fibers are pulling 23 chromosome towards the south pole, in the sense another 23 is moving towards the north pole of the cell, okay. So how the chromosome looks like, in the sense the chromosome looks like after pulling, it looks like this, okay. So this phase is called as anaphase 1. What is this called? Anaphase 1. Just imagine. So how it looks like, in the sense, It's like this. Okay, I'll show the chromosome. This is the homologous chromosome, and this is another pair of homologous chromosome. Okay, this stage is called as anaphase. Anaphase 1. Okay, so how many chromosomes are moving to the south pole? In the sense, it's only 23 moves towards the south pole, and another 23 moves towards the north pole. Now imagine and tell me what is happening here. The chromosome number is just divided into half. Yes or no? So this is the nucleus. In the sense, nucleus, see, after the anaphase it is called as telophase. So what is happening in the telophase? The nuclear membrane reappear. Okay, the chromosomes are also starts to become a chromatin fiber, isn't it? Isn't it? So how many are here? 23, and here 23. Okay, this is called as telophase 1. Telophase 1. This is anaphase 1. So what is happening in the anaphase 1? Anaphase 1, the chromosome number is divided into half. Okay, divided into half. So at the end of meiosis 1, how many cells are forming? There are 2 cells. Okay, how many cells are forming? In the meiosis first, at the time of telophase, it divides into 2, and each one consists of how many chromosomes? 23 chromosomes only. But at the stage of meiosis 1, it is prophase. If you take in the prophase, how many chromosomes are here? It is 46. Okay, 23 pairs are here. But at the end of meiosis 1, it is completely reduced into half. Okay, so this 46, that is 23 pair. Okay, 23 pairs are considered as 2n number of chromosome. Okay, 2n number in the sense 2 sets of chromosome, 2 sets of homologous chromosomes. So how many are there in the human being? It is 23 pairs, that is 46. Okay, when at the end of this meiosis 1, what happened? There are two daughter cells. They have n number of chromosome, that is only one set of chromosome are present. Clear? Coming to the next one, what is that? Meiosis 2. Meiosis 2 also contains 4 stages, that is prophase 2, metaphase 2, anaphase 2, and telophase 2. Okay, so what is happening here? In the sense, just like that of which one? The meiosis 1. But here what is happening? At the end of meiosis 1, there are 2 daughter cells, isn't it? There are 2 daughter cells with n number of chromosome we have seen in the nucleus. Now this is entering into which phase? Meiosis 2. Entering into meiosis 2. Okay, so what is happening in the meiosis 2? In the sense, just like that of the mitosis. So what is happening in the mitosis? What happened here? See, just we take let me take another color. So what we take in the sense, just we take 2 chromosomes. Okay, so here 2 chromosome, here 2 chromosome we take in the sense, in the time of metaphase, this nucleus, if you take, what is inside this chromosome? Okay, sister chromatids. The chromosome divides into chromatids. Here also chromosome divides into chromatids. Okay, and now nuclear membrane starts disappear. Okay, which one disappear? Nuclear membrane starts to disappear. Okay, this is the chromatid, and this is the chromatid of another. Okay, now the nuclear membrane starts to disappear. So as a result, what happened? As a result, here the centriole in the sense, here also centriole, and the spindle formation occur. Okay, spindle formation occur, and in metaphase the chromosomes comes to equatorial plane, and then the spindle fiber starts to contract. Where the spindle fiber attached? At the centromere. Okay, centromere. So again, just like mitosis, one chromatid comes to this side and another chromatid moves to the another side, opposite side. Okay, so at the end of the telophase, we get how many cells? We get 2 cells. Okay, 2 cells, one with the number of chromosome will be same. The number of chromosome will be same. The sister chromatid synthesize the another chromatid of the same nucleic material. Okay, whereas here the nucleic material, the nuclear material will be differ because crossing over takes place, isn't it? So this cell get one type of nucleic material and this is another type of nuclear material. Whereas in the second division, the nuclear material of this cell is as same as that of the next one. Okay, so both are same. But at the end of meiosis, I told you already we got 2. The 2 is again divided. That is, this one divided into 2 cells and this one again divided into 2 cells. Okay, the same pattern takes place. Same pattern takes place in this side. Now I explained in this side. So how many are at the end? 2. Here also 2. So totally how many cells formed? How many cells formed? Totally 4 cells at the end of which one? Meiosis. Okay, at the end of meiosis, see for example, this is, let us take meiosis 1. At the end of the meiosis 1, we'll get 2 daughter cells, isn't it? 2 daughter cells with n number of chromosome. Okay, n number in the sense 23. Here it is 23 and here it is 23. Okay, and now this undergone the metaphase 1, anaphase 2, metaphase 2, anaphase 2 and then telophase. At the end of the telophase, we get 2 cells. Okay, here also the same, metaphase 2, anaphase 2, then telophase 2. At the end, it forms 2 cells. Okay, 2 cells of n number of chromosome. N number of chromosome in the sense 4 daughter cells are formed at the end of the meiosis 2. So the meiosis division, at the end of the meiosis, single meiosis division, how many cells we got? We got 4 daughter cells of n number. So just imagine, where is this division occur? Just imagine and tell. Where this division is called as reduction division. Okay, why it has to happen? Why it has to happen and where it is happening? Just think over and tell. Just think over. Where is this happening in the sense? Why it has to happen? First, let us think about. Okay, so I'll explain where it is happening in the sense. It happens in the male as well as female sex organs. Okay, male as well as female sex organ. So what is the male sex organ? It is called as testis, right? And the female organ is called as ovary, right? So the ovary produce ovule. The ovary produce the ovule and this testis produce the sperm, right? Sperm. So the fusion of these two are called as zygote. Okay, this is called as zygote. Now see, I told you reduction division in the sense n number of chromosomes. So the n number of chromosome formed in where? Ovule. Okay, ovule. This cell is an n number of chromosome cell. Which one? Ovule. Ovule is a n number of chromosome cell. At the same time, this sperm is also contains n number of chromosome. Okay, so why it has to have the n number of chromosome in the sense, at the time of fusion I told you the homologous chromosomes, which comes 23 comes from father and 23 comes from mother, right? So while fusion, these two nucleus joined. Okay, so there is a nucleus in the sperm also. So the nucleus will be released inside this ovum, and these two nucleus, they join and they form the zygote. Okay, after fusion, this is called as zygote. So how many number of chromosomes are here? I told you here it is 23 and here it is 23. Okay. So how many are here? It is now 46. Now we understand why the reduction division is necessary. Reduction division is very, very essential. Okay. Why? In the sense, then only the new human being form. Okay. Otherwise, if there is no reduction division, just imagine this contain 23 pairs, that is 46 chromosome. And if this also contain another 46 chromosome, so total how many chromosomes are there? 92 chromosomes, right? So 92 chromosome, in the sense it is not a human being. Human being consists of only 46. If it is 46 only, then it is a human being. If it is 92, it is not a human being, it will be some other organism. Understand now? So reduction division is very, very important for what? The production of another human being. Okay, another human being. So this is very, very important question. It will come in the NEET examination. So which division is called as reduction division? In the sense, it is meiosis. Why it has to happen? In the sense, at that time only the 2n number of chromosomes are reduced to n number of chromosomes. Then at the time of fusion of the ovum and the sperm, then the zygote form with the 23 homologous pairs of chromosome. Understand? Now we understand. So 23, this 23, the size and shape are same. Okay, same, but the genetic materials are different, right? Okay, very good. Now I told you why the meiosis is very, very important. So meiosis, cell division takes place only in the sex organs to reduce the number of chromosomes in the sperm and ovum, and this is the very, very important point, as well as the significance of the meiosis. Okay, the genetic material, the number of the chromosomes will kept same, as well as the variation in the human beings shown only through the meiosis. Why? In meiosis only, which will happen? The crossing over of the non-sister chromatid takes place. Yes or no? Yes or no? So this part I already shown you how the crossing over occur. So this chromatid joins with this part and that chromatid joined with that part. Okay, so here the genetic materials are exchanged. Because of the exchange, there is a differentiation between one generation to another generation, right? And one generation, that is grandparent to parent, parent to children, and children to another, their children, there will be a few variation because of this one. Okay, in their intelligence, in their morphology, and in their physiology, everything will vary because of this one, and their mental ability, everything will be differ because of this exchange. Okay, so the crossing over only occur in the meiosis, right? Let us see a small part of this in the form of, okay, how this is occur. In the sense, I told you it is already meiosis 1, prophase, metaphase, anaphase and telophase 1. Then meiosis 2, if you take, it is prophase 2, prophase, metaphase 2, anaphase 2, and then telophase. Okay. Let us see the meiosis 1, how it happens. So you can see what you are seeing. This is a diploid cell. Diploid in the sense 2n number of chromosomes will be there, and these are the chromatin fiber. They are condensing here. You can find how it is condensing. Okay, and this is the homologous chromosome. See, this is chromatid, sister chromatids. You can see the variation. Now these two are the homologous chromosome and these two are the homologous chromosome. Can you find the size as well as shape. Now it is okay, they are come closer. This is called as tetrad. Okay, tetrad. You can see the genes, how it is arranged, as well as alleles. These are genetic materials which are present in the chromosome. At the time of cell division only we can see the chromosome. The resting stage, they will be the chromatin. You can see the crossing over. Okay, crossing over occurs. This is a recombination of the chromosome in different see, I told you this has the nucleus and this also. So the variation happens because of this. Okay, the crossing over. Coming to the few questions I am going to ask you. See the first question. The end of meiosis, dash number of cells are produced. Give the answers very quickly. Yeah, very good, very good. Everyone answered. It is 4 cells. And meiosis is a dash division. I told you which division it is called as. Yes, yes, yeah. Few members not answered. It is reduction division. Reduction means what? The 2n number of chromosomes are cut into n number of chromosomes. Okay, 23 moves towards one cell and another moves towards another cell in the which division? Meiosis 1. Coming to the next one. In meiosis division, dash number of chromosome cells formed. This already I told you here. It is n number of chromosomes. Okay. Coming to the next one. Meiosis division takes place in dash cells. Dash cells. In reproductive cells. Which cells? In reproductive cells. In female it is called as ovary and in male it is called as testes. Right. Coming to the next question. Crossing over occur in dash stage of prophase. Dash stage of prophase. Which stage? Very good. Quick. Yeah, it is packaging. Very good. Everyone answered. Coming to the next question. What is that in the sense? Name the third stage of prophase in meiosis. Third stage of prophase in meiosis. In which stage the crossing over occur? In which stage crossing over occur? You can see in the previous question, is right. What is that? Packaging. Very good. Yeah, few have answered. So make a note. Okay, crossing over occur in which one? Packaging stage. So that is the third stage of prophase 1. Okay, very good. So thank you each and everyone. And this class is very important for you. And you share this video to your friends, those who are appearing for the NEET examination 2025. Okay, thank you so much.